Thank you, Amy. I hope everyone can hear me. Good morning. My name is Pawel Mroz. I'm assistant professor here in laboratory medicine pathology. And I'm going to talk today about the implementation of pharmacogenomic testing program at MHealth Fairview and beyond. Well, that was the title that I had for the purpose of CME, but the actual title is Trials and Tribulations of Young Naive Molecular Pathologists Attempting to Implement PGX Testing Program, or the Myth of the Low-Hanging Fruit. My objectives for today is to describe a successful and highly collaborative implementation of pharmacogenomic testing, outline the approach, development, and validation of the in-house PGX assay, introduce Go for PGX software tool for genotype-phenotype translation, and gene phenotype drug CPIC-based recommendations, discuss clinical workflow that connects the providers with the PGX experts and the molecular diagnostic lab and pharmacy, and present efforts to create a statewide clinical decision support review committee. I do not have any financial disclosures. I do have one disclosure. I have two parakeets in the house that occasionally become very loud when I'm on Zoom. So I'll try to put them in the place that they should not be heard, but if they manage to be heard, I apologize for that. And I want to start with a summary, because I've noticed that I have a tendency to lose my audience along my presentations. So I think I'm just going to give you the summary as a map where we are. So the project started as any project does, with buckets of enthusiasm somewhere up here. And it follows a kind of curve like that. So when we start with buckets of enthusiasm, we feel empowered to do great things. And I think that's how many of the great artists and architects start. And I'm neither great nor an artist, but I had a lot of enthusiasm when sometime in June 2019, Bharat approached me and said, why don't you develop a pharmacogenomic testing? And I'm like, well, I don't really know much about it, but sure, why not? Hold my tea. I'll get on with it. What I was really doing, I was blindly walking over a cliff of steep learning curve. And I always thought that the steep learning curve is the place when you're climbing. But what I've learned in the process of this project is that this is the steepness of the slope that you're tumbling down because of lack of knowledge. And you end up in a moment of that Socrates had a long time in the ancient Greece, when you realize that I know nothing. This is obviously not Socrates. This is a Manuel from the British sitcom Fawlty Towers, and he was Spanish, and he could only say, I know nothing, and I'm from Barcelona. So I very quickly, during the process of this project, arrived to that moment when I realized that I know absolutely nothing. And then I had to look up and figure out the slope of accomplishment, and realize that the path to accomplishment of this project is going to be very tortuous and treacherous, and there's going to be a lot of problems to solve. So to do that, I enlisted help, and that's how the Fellowship of Pharmacogenomic Testing began. And we soldiered on. And then we tried to define the progress, and the progress initially was very slow. It was almost like this snail in the math exercise that, at least when I was a kid, we all had to solve when we were told that the snail climbs 2 inches of a tree trunk per day and then slides down 1 inch during the night, and then we had to calculate how many inches the snail will climb up over the week. Obviously not far, but then that's how we define progress. So this is roughly how the whole project, if I were to graph it, went on. And now I'm going to move on to the details of it. So the steep learning curve. Well, first, what is pharmacogenomics? Well, pharmacogenomics is a study of genetic determinants of drug that are responsible for different responses of medications by individuals. There are pharmacogenomic variant alleles that occur in genes that code drug metabolizing enzymes and alter their enzymatic activity. And depending on the genetic makeup, some drugs may work more or less effectively for some patients than they do for the others. And some drugs may also produce more or fewer side effects in some patients than in someone else. So the sort of overarching goal and promise of clinical pharmacogenomic testing is that we will get to the place where we will use genetic information to effectively deliver the right drug to the right patient at the right dose. And pharmacogenomics has been around for a while. The initial discoveries that there are enzymes and genes responsible for medication metabolism go back to mid of 1970s. But it's been the last decade or the last decade and a half when the pharmacogenomics really became this new and transformative science and healthcare paradigm change. And there is a reason for that, because there's a growing evidence that the genetic variation within genes that are involved in metabolism or mechanistic pathways of medications is very important and able to predict therapeutic response and toxicity, ergo help us to avoid adverse side effects. And in terms of numbers, we now know that at least 90% of patients carry one genetic variant that require medication adjustment, and 50% of patients carry 5 or more genetic variants that may lead to medication adjustment. And that between 25 and 90% of variation response of medication is in fact due to the genetic composition of patients, and that over 90 medications across classes with CPIC dosing guidelines are based on PGX results. So when we talk about pharmacogenomics, what we're really talking is a big family of cytochrome oxidase P450 enzymes, and this is something that we all kind of learned briefly in med school, and then conveniently maybe forget, just remembering the P450. But it's a very complex family. It has 57 different active genes, 17 different families, and it has this very complex nomenclature. So that nomenclature is very confusing because it was developed by the researcher who discovered the first family member, which is called CYP2D6. It was in 1976 when the first published paper about CYP2D6 and metabolism of drugs was occurred. And that person then gingerly went on and developed the whole nomenclature for pharmacogenomics enzyme that has nothing to do with genetics. And you can't blame him. The human genome nomenclature, the first guidelines were published in 1987. So Mary Levenley's years later, so he didn't really have that. So what he came up with is we have a cytochrome P450. This is a superfamily, and then he gave family. There are numbers of different families. So this is family 2, subfamily D, this is isoenzyme 6, and then star allele is the actual allele variant that we can detect by genotyping. It's complex, it's unwieldy, and not very good, but it's been embraced, particularly by the pharmacists, and this is what we have to work with. So that was all part of that learning curve. And then on top of just understanding the genetics implications, there is also the fact that the pharmacogenomics work in pair with as a gene-drug pairs. And there are clinical guidelines and clinical societies that have been developing those guidelines. There is Clinical Pharmacogenomics Implementation Consortium. This is the US-based nonprofit organization that is releasing periodically clinical guidelines about the gene-drugs interactions. Along the way, PharmVar came along. This is a massive database that holds most up-to-date definitions of all particular SNPs that define each of the star allele for several of the genes. A couple of years ago, US Food and Drug Administration got into the pharmacogenomics, and they released a complete, in their mind, list of about 200 medications that are affected potentially by pharmacogenomic testing. And there is also a European or Dutch pharmacogenomic working group that they've been also releasing their own guidelines. And this is a Venn diagram that how often they actually been agreeing. So there is a lot of disagreement what means what. So to deal with that, about 2013, a farm GKB evolved from a collaboration between CIPIC and DPWG. And this is the place that and the experts that took upon themselves to unify the pharmacogenomic clinical annotations and clinical guidelines to define very important pharmacogenes and things like that by meticulously peer reviewing published literature, including other databases, and trying to make a sense of all sort of coexisting guidelines. So this is what we currently try to use. So there was a lot of learning up front, and I will admit that pharmacogenomics is not a big part of testing when I was a molecular fellow. It was only starting, and maybe there was like 1 or 2 questions about pharmacogenomics on molecular board. So obviously I got pretty quickly to the point that I realized that I know very little to nothing about pharmacogenomics. But I had to figure out how to start on that path of accomplishment so I can actually do something here. And the first thing was the selection of testing platform and assay development and validation. So I decided to play to my strength. I am a molecular pathologist. This is what I do. We develop and validate molecular assays. And working in the molecular diagnostic group laboratory, I want to mention 2 people that came to my rescue, and that was Stephen Michael and Teresa Kemmer. They've been fantastic and very helpful in the whole process of bringing this testing together. So there are certain principles to develop the PGX assay. A well-defined designed assay must identify all significant variations that have an impact on drug metabolism. There are different methods that can be applied to analyze a patient's PGX profile. And the selection of appropriate technology must be based on the rigorous evaluation of several factors like sensitivity, specificity, sample requirements, and costs. And since different SNPs may signify different outcomes, the SNP composition of the assay is also extremely important. And then we have to look at the feasibility of implementation and particularly of the cost. So looking at the types of platforms and assays that were available at that time, the most commonly used one is PCR targeted genotyping. And this is the type of method that we decided to go with and validate, mostly because this is the most cost-effective method. Pharmacogenomic testing currently is still poorly reimbursed and it's mostly out of pocket. So we were trying to keep the testing costs relatively low. Now, there are other methods that are bigger and like SNP microarrays. They are used by some of the laboratories. They open the opportunity to test thousands of genetic targets, many more targets that currently have clinical guidelines. But they are complex and more expensive. And those chips, once they are produced by manufacturer, they're not very easily customizable. So if there is a new update coming up, and you have the validated assay, it's not easy to change that. And then there is next generation sequencing. And we can do whole genome and whole exome sequencing and pick up all sorts of different genetic variants. But those approaches are currently still too costly and prohibitive. But there is also next generation targeted sequencing. And this is something that I will talk about a little bit later. Now, how do we know what alleles to actually put on the assay? Well, AMP realized that there is a lot of discrepancies between different assays that were or are on the market. They did the study for CYP2C19 here, and they looked at different commercial assays that offer pharmacogenomic testing, and they looked at the different star alleles composition. And as you can see, Even the ones that are FDA cleared, they have, the bare minimum of the alleles, but there's many more alleles that are important in CYP2C19. So I'm slowly been working on releasing guidelines and recommendations for a minimum set of alleles for all genes, but it takes time. The way AMP works, they've been able to release guidelines for 1, 2, 3, 4, 5, 6 genes. The next month there are 2 additional genes that are going to be published, CYP3A4 and CYP3A5, and then there is another guideline expected later this year for DPYD. So that will bring the total to 7, but there is 17 CYP genes that are already required for testing. So this process is slow, and it doesn't allow us to rely solely on it. And why is the composition of the assay so important? Well, it's important because of the convention and the nomenclature. So if the one laboratory does not test for certain SNP, like in this case, does not test for the SNP that defines star 17, it will by default report the patient as the patient is a wild type. And the wild type in pharmacogenomic nomenclature is designated as star 1. So if this laboratory is not testing for SNPs defining star 17 for CYP2C19, the report will say that the patient is a normal metabolizer. If the other laboratory has a SNP that defines star 17, this laboratory will interpret the result as a rapid metabolizer. So there's a difference in interpretation depending on how the assay is put together in terms of the SNP composition. So what we did, we reached out to College of Pharmacy. We tried to follow AMP-specific guidelines. We also talked to our Fairview pharmacists, and we also reached out to other institutions that have successfully implemented pharmacogenomic testing. And we put together our Go4PGX assay that currently has 9 genes, 32 SNPs, and 32 star alleles. And we also have a separate assay that allows us to figure out copy number variation for CYP2D6. And we are able to provide recommendations across 33 different medications, across 20 therapeutic classes, potentially aiding 8 clinical specialties in how they distribute the drugs to the patients. And we validated our assay for both peripheral blood and buccal swabs, and our expected turnaround time is about 3 to 5 days, depending on when the sample arrives to the laboratory. And we currently bill it out of pocket. So that was just the first step. And then once we did that, I realized that I'm going to need a lot of help from others to get it to the stage that we can actually successfully report results from genotyping. And fortunately, Bharat again sent my way an opportunity to apply for Minnesota Learning Health System scholarship program, and I was fortunate to be selected to be one of the scholars. And I would highly recommend it to any young pathologist that is looking for an opportunity to get a career development award, because the purpose of this program is to train clinical and research scientists to have the skills to support their efforts to apply patient-centered outcomes research. But they also, do research in rapid implementation. And I always think that pathologists are natural implementators. This is what we do. We develop assays, whether this is IHC or molecular or some other testing or chemistry assays or implement processes that allow us to better diagnose the patient. We kind of have those natural skills. What this program offers is an opportunity to talk to people that actually understand the science behind the implementation and learn about the ways to evaluate the success of implementation and patient outcomes and things like that. And what this program allowed me to is allowed me to put my pharmacogenomic implementation efforts into actually specific aims for the scholarship. and also put me together with with other scholars that are doing implementation science, but also opened up opportunity to talk to other scholars that are recipients of career development awards across the whole University of Minnesota. And this is a very, very interesting collaboration. It includes, School of Public Health and the University of Minnesota. It has collaborators from Hennepin County Medical Center. We get, introduced to collaborators from Mayo Clinic, and we participate in Chem Scholars program. And there are several other partners. So that frees up a little bit of your academic time. And it allows you to work with people that know a lot about implementation and learn from them. So once I was fortunate to qualify for that program, the next step was this. We wanted to select and develop a validated PGX reporting platform. And to do that, I teamed up with Institute of Health Informatics. And this is why. At the top here, this is how the results may look like when we take them off the genotyping instrument. We have a selection of SNPs that we're testing for. We have a case here, and we have essentially a nucleotide change. This is absolutely useless from the perspective of a clinician. They cannot really use that for anything. So the question now arises how we take those genotyping results and we translate them into a readable report that the busy clinician can actually see and use in the EHR. So there are commercial options, and we initially looked to commercial options. And I've met with several different vendors and interviewed them. I actually developed a structured interview process with a series of questions that I posed to every vendor representative. And we created like a large spreadsheet to compare the responses. And then I also reached out to the Institute of Health Informatics to see if there may be an interest in developing the in-house reporting tool. And one of the reasons why we decided to go with the in-house is that there's been emerging data suggesting that the commercial vendors may sometimes overcall some of the phenotypes on pharmacogenomic testing. And there is a recent paper that came out that actually looked at several different commercial vendors, and they compared their interpretation of the variants to the actual CPIC phenotype guideline interpretation. As you can see, there is significant difference across the interpretation. So, and another reason to go with in-house development was that we had a meeting with Fairview, and the cost was felt of hiring the external vendor was felt at that time to be prohibitive. So we designed a reporting software. We called it Go for PGX to provide PGX test results. And that software produces a PDF report that details the genotypic data and the phenotype translation for all the designated genes that we have on our PGX testing panel. And we got a little bit of a breakthrough along the way because CPIC happened to build the database of their guidelines. So until a certain point in time, all CPIC released were large Excel-based spreadsheets, and we were prepared to build a big database of Excel spreadsheets and then work off that. But fortunately, a couple of months into the project, we realized that there is a database available for download from CPIC, and that definitely streamlined the process. So our software works as a sort of a web-based software. It allows the technologists to upload and import the genotyping data from the genotyping instrument. The software will separate each case separately. It will allow us to review the results, preview the report, and do the approval. It will also flag if there are any problems with diplotype issues, like if the algorithm cannot call some of the diplotypes. We can go into each individual case and review each individual gene and see the diplotypes being called and the phenotype being interpreted. We can then review each individual gene within the software and review all the calls that are being made for every single individual SNP. The software does all possible phase combinations, and it allows us to either automatically call the diplotype, or we have a custom override option if we disagree with, or if the software cannot call the diplotype. And then it builds the report. The report is then manually downloaded back into the molecular diagnostic lab and then uploaded into Epic. And the end users can find the report under the title Pharmacogenomics Profile in the chart review. And then the report has 3 sections. It has PGX results with the specific genotype calls that we can trust, because now we automatically use the CIFIC database to make those calls. It has recommendations organized by medication class. This is again based on CIFIC supported recommendations. And it has all the description of PGX variants with patient-specific SNPs and variants that we tested for listed in the report. So if the patient wants to go to a different healthcare system or a different provider, they will have all that information available. So once we have that, we started working on developing ordering and testing workflow. And with that, we teamed up with Fairview Pharmacy. And we decided to build on an existing infrastructure. So clinical pharmacists have developed a process for send-out pharmacogenomic testing, and the molecular laboratory had an existing germline testing pipeline. And I wanted to take advantage particularly of our existing germline testing pipeline, meaning the availability of genetic counselors. We have fantastic genetic counselors here. We have some that we work daily in molecular diagnostic lab, and some that are also working solely in the clinics. And they are well positioned to talk to patients about and discuss the risk of genetic testing. So I insisted that when we build our workflow, it will start with the genetic counselor. So once the order is placed for pharmacogenomic testing in a smart set, it will generate the genetic counselor consult with the patient. The genetic counselors will fill out forms, and that will and did improve the submission of the forms and alleviated some of the burden that was previously put on the clinicians, and improved our turnaround times. Then the testing is done in the molecular diagnostic lab. And again, as I mentioned, we can receive 2 types of specimen. The test can be added to any peripheral blood draw, or clinics can do the buccal swabs, or even we can send the buccal swab kits to the patient's homes and receive them back. And once the testing is done, go for PGX will generate the report, and the report will be appended into Epic, and the molecular pathologist signs it out. And then that creates a pharmacist NTM visit. And the pharmacist will put the final recommendations into a note. And the reason to have that is the pharmacist at the end of it is that pharmacists are uniquely skilled in identifying drug gene interactions and other interactions that, with medications that the patient may be taking. And they are the last pair of eyes before the final note that they create goes to the originating provider. So we've created a smart set within Epic, where the providers can now find our Fairview PGX testing, and it is connected with the referral to GC testing, to GC visit, and that's how the whole workflow starts. So once we've created all that process, we needed to reach out to the providers and offer them education about the assay and also about the pharmacogenomics. So we started with educational sessions that were targeting high-use clinics and providers. These are the ones that we identified that they were generating high volumes of send-out testing. And we did targeted phase 1 education. We did presentation on the presentations of the in-house testing and comparison of why to use in-house versus commercial tests with about 10 rounds of sessions across primary care and behavioral health. We did system-wide education approach. We had neighborhood news articles. We have intranet articles. There is a SharePoint site created with info about pharmacogenomics. I was invited to give the Fairview ground rounds. Our pharmacists gave Fairview ground rounds. So there's been a lot of system-wide attempts to educate the providers. And then we also had to create a pharmacist education process. So until recently, Fairview did not have a single PGX fellowship trained pharmacist. We now have one. Tiana Butler is a PGX fellowship trained pharmacist, but our MTM process, so the end users are pharmacists, are not PGX trained, and we've reached out to a group of approximately 15 MTM pharmacists and created sort of a basic training about basic genetics, pharmacogenomic resources, and CP guidelines. So while we have Tiana, who's kind of overseeing the whole process and is the contact person for tough questions, we are relying actually on MTM pharmacists and the training that we provided to use our report and provide the final pharmacogenomic notes. We also teamed up with College of Pharmacy. They created a PGX Echo program. It's an interesting program. It's almost like pharmacogenomic tumor board when one can submit a difficult case, and then there is a once a month PGX Echo meeting when pharmacists and other pharmacogenomic experts join via Zoom from several different institutions across Minnesota and sometimes beyond. And one can discuss the identified patient case with the results, and they can one can hope to receive some additional input from other pharmacogenomics experts. And I've participated in several of those ECHO meetings, and it's fantastic educational opportunity for me, but it also offers opportunity to discuss some of the complex cases, both at the genetic and the medication level. And this year, I was also, actually last year, I was also invited to co-organize the pharmacogenomics conference here at UMN. We got over 300 participants across the country. It was a virtual 3-day conference, and it's going to happen every 2 years. So our providers and pharmacists have an opportunity to listen to the national experts in pharmacogenomic testing and implementation of pharmacogenomics and learn from there. Now, beyond what we've implemented right now, there is this idea of developing clinical decision support, an active clinical decision support that will interact with providers at the time they are trying to prescribe the medication that may be either affected by the already existing pharmacogenomic result, or the medication that is required to get pharmacogenomic testing. So this is an effort that has been spearheaded by College of Pharmacy, and we've joined that effort as a molecular diagnostic laboratory, and also Institute of Health Informatics is working with us to provide the backbone of IT support for this effort. And this effort started and is spearheaded by Dr. Pam Jacobson from the College of Pharmacy as the University Grant Challenge Award that aimed at implementation of PGX at state scale. And I was drafted in that statewide clinical decision support committee. And what that CDS content review committee does, it aims to create the governance structure and develop standardized process to transform evidence-based guidelines into shareable pharmacogenomic clinical decision support. And we have a small working PGX group that initially met weekly. I think now we meet every 2 weeks. And then we have a statewide committee with members from several different healthcare systems. So apart from MHealth Fairview and College of Pharmacy, we have members from Alina, Essentia, we have members from Sanford and Mayo, and we have occasional guests from North Shore in Chicago and other friendly programs. And the idea behind this statewide content review committee is twofold. One is to create go for PGX content that will eventually go into the CDS alerts that will fire within the EHR. But the other idea is that we will create it as a shareable content. So it's not only that MHealth Fairview can implement it, but any other participating healthcare system may tap into those resources and use that information. So we're trying to leverage the enormous amount of intellectual power across the members from different institutions to sort of divide and conquer a lot of work that needs to go into creating a CDS alerts, because there's lots of genes. Each gene has many different phenotypes. Each phenotype has a different comment, and there are also different drugs that we need to tackle. I mean, the FDA list of drugs that are affected by pharmacogenomics is 200. So we're creating both gene and drug monographs. Those monographs are reviewed by the experts, and then the committee meets and votes on the specific verbiage for essentially each section of the monograph. It's a lot of work. We've done about 15 medications so far and about seven gene monographs. So it's a slow process, but it's definitely worthwhile. And the idea is that eventually we'll be able to build an active PGX CDS for Epic that will fire this kind of alert. This is an example for the clopidogrel prescription. Clopidogrel may be affected by CYP2C19 patients that are poor metabolizers, and those should be considered to have an alternative medication, other P2Y12 inhibitor like Pathology. Something different. So this is an example of the possible future CDS. So how do we define the progress? Well, today is actually one day after a 1-year anniversary since we went live. So we went live on April 18th, 2022. And for the first couple of months, we had very few samples, which was good and bad. I mean, it was good because we didn't get overwhelmed with initial volume of testing, so we could figure out some of the problems that may arise. The bad, obviously, is because we wanted to start strong. Now, one of the reasons, looking back at and sort of analyzing what's been happening here in those early days of testing, is that we relied on GC visits, and maybe we did not communicate it well. So one of the things that was happening is that there were not enough appointments for GCs available to meet with patients tested for pharmacogenomics. So that created a bottleneck of testing and slowed us down. But the other was a glitch in the way we created the smart set for ordering, because we left out the option for providers to decline the GC visit. And some of the providers went ahead and declined the GC visit, and that put the patient and the sample completely in the limbo, because then it completely messed up the workflow. So we had to change the smart set, and now the smart set has an embedded GC visit, and individual providers cannot skip that. And now we also have a streamlined process for setting up the GC visit. They're seeing patients on certain days, sort of in concordance with the PGX clinics. They can see about 5 patients in 4-hour blocks. And we are also streamlining and bringing together GC and PGX calendar to kind of help with the volume and alert them when the ordering is happening. Now, we still face the competition from commercial vendors, and that's a big problem, not only for pharmacogenomics, but for any genetic testing or probably other testing that is offered by commercial laboratories. Part of it is habit. There are certain providers that are used to certain vendors, and part of it is the fact that the commercial vendors are very effective at reaching out to providers. So there are several commercial vendors that are competing in this space. The biggest one is GeneSight, and we are actually succeeded in convincing Fairview leadership to turn off the pharmacy support for GeneSight reports, and that happened in August. Nevertheless, GeneSight offers the ability for the providers to set up their individual accounts, sort of like you set up your Amazon account and then do the ordering. So we still, see, a huge number of tests in 2022. We saw 370 orders that were placed to GeneSight, and One we saw about 38. One is driven mostly, the One volume is driven mostly by the BMT clinic, and that's because we don't have 1 gene and 2 SNPs on our assay that they want to use, so that's why they're sending it to One. We've been able to curtail it a little bit. I mean, this is 2023 up until April, so 4 months. If you multiply by 4, it's going to be less than 370. So I think we are convincing some of the providers to switch to our assay. This is the profile of the clinics. This is kind of bittersweet. So Uptown and Lakeview and Egan, these were the heavy users based on the prior send-out testing, and we targeted a lot of education towards those clinics. As you can see, we were not that convincing. But we've convinced other clinics, including UMC and several other clinics, successfully. So we gained new users of our in-house testing, and maybe that's reasonable. These are the users that may not have been previously either using pharmacogenomic testing or may not been wedded to the certain ways that they order pharmacogenomics testing. So that's a success story. But it's a little bit disappointing that we cannot convert more of the heavy users that are probably contributing to that higher number of send-out testing. We're working on that. We're working with them, and we're hoping that that this is just a temporary situation. In terms of providers demographics, we see about half and half of MDs or DOs and nurses or PAs ordering the pharmacogenomic testing. But I think this is the interesting breakdown. So behavioral health or psychiatry in general, they've been at the forefront of implementation of pharmacogenomic testing from the very beginning. They are the ones that spearheaded the efforts of bringing pharmacogenomics into the clinical implementation and generating lots of literature that is out there about pharmacogenomics. But we're not seeing them as the heaviest users. Maybe these are the ones that are using GeneSight. In fact, I know that one of the behavioral health clinics straight out refuses to use our test. We're talking to them and trying to educate them, and there's been few breakthroughs done, but initially they were just saying that they want to use our in-house testing. Now, what is quite pleasing is that the majority of the testing that we get is from family medicine. So primary care providers, they also take care of a lot of behavioral health patients, but they also take care of other patients that we provide pharmacogenomic interpretation for the medications that those patients are taking. So within the family medicine, we've been very successful in convincing them to switch to our assay. Now, looking at some of the patient profiles, what we've seen so far is that the majority of of the patients that are receiving testing are between 18 and 50, which is kind of interesting and a little bit counterintuitive because in principle, the biggest use for pharmacogenomic testing should be those two groups because these are traditionally the groups that are on a polymedication. The younger population tends to be usually on one, maybe two medications, at least based on statistical data. So the fact that we're seeing a majority of the patients in this younger population probably suggests that they are on a very specific medication and the providers are using pharmacogenomic testing to fine-tune the single type of medication. I don't have a good explanation why we're seeing the predominance of female patients here. We're looking more into that data to clarify. This is sort of data taken out from Slice Advisor, but I have an IRB pending to do a full scope project to evaluate the 1 year of pharmacogenomic testing. We want to put out a paper, like a follow-up paper to our validation paper describing all the processes, including the patients and the medications that we provided recommendations for. But in terms of some of the clinical complaints, the patients identified a lot of it is adverse drug episode of the adverse drug reactions. Significant proportion is depression. And if you look at the percentages, obviously the patients present with more than one, so there is overlap here. And the vast majority of patients were identified as white. We're again looking into it to see how those percentages compare to the demographic profile of Fairview patients or individual clinics, and we're going to try to include that in our follow-up paper. So this process that we went through, it seems like never ending because we are now here at 1 year anniversary of our testing with our. We think we've figured out most of the problems with our clinical and testing workflow. We're working on education. We're increasing the volume, but now then we're back to the beginning. So sometime along the way that we realized with discussions with pharmacies that our initial assay, while very good, is not optimal. We're missing some of the clinics that would like to use our in-house testing, like BMT clinics. With bigger, assay, we may be able to convince some of the reluctant users. So we look back again at the technologies that are available, and we were thinking initially, well, maybe we just expand our multiplex PCR-based assay and include additional genes and SNPs. But that quickly became a very complex project. So we decided to see if we can switch to the technology that will allow us to do much more, and that's targeted NGS-based genotyping. So right now, we're in the process of validating NGS-based assay, and we're going to go from 9 genes to around 40 genes, including 17 CPIC genes. So we're going to keep some of the additional genes validated because we know that CPIC is working on some of the additional genes beyond the currently 17 that they have on their website. And we're going to go from mere 32 SNPs across those genes to over 2,000 different SNPs. And that was a massive work to create like the bad file for bioinformaticians, because some of the SNPs have defined different star alleles. Some of the SNPs define star alleles that are currently of unknown function. So we will not be reporting those, or we will be getting information because we'll be sequencing all the genes. So all the exons across all those 40 genes, as well as selected intronic positions that define specific star alleles for those genes. So we'll be getting a lot of information, and then we're going to be working with MSI bioinformaticians to trim it down to use for the report only the ones that have a known function. Now, this is combined or in parallel with working now with Fairview IT and with Dr. Michelle Stoffel to develop and implement active CDS. So we want to go. So if once we switch to that large panel, the PDF will become not the best way to report the information. We still want to keep it in case the patient will not travel across different healthcare systems, but we want to use this opportunity to build an active CDS within the Epic. So we've been working with Fairview. We've secured the ability to license the genomic module, which is a portion of the Epic that allows us to put discrete genomic data into the Epic. And we now have resources secured within the Fairview IT with a project manager. And the project is twofold. It's to build the Go4PGX Fairview interface so we can automate sending the data into the Go4PGX and receiving the data as discrete values together with city-based recommendations, and then put them back into the genomic module, and then use genomic module to create active CDS for any medication or any drug and that is affected by pharmacogenomic testing. So the clinicians, when they attempt to prescribe that medication, they will be alerted that there is a pharmacogenomic result and there is a pharmacogenomic-based recommendation that affects the dosing of the drug that they are about to prescribe. This is what we're going to be working in the next months to a year. We continue to reach out with educational efforts to providers and also now to patients. College of Pharmacy has created a website for patients. And we're trying to see if we can utilize that website. We're also working on another smaller assay. This is one gene assay, CYP2C19 assay, specifically designed for clopidogrel. We reached out to the cardiology clinics that are doing interventional cardiology for patients that are presenting with acute coronary events, and they're going to be undergoing PCI. Those patients are initially prescribed clopidogrel, but if they are poor metabolizer, those patients may not benefit from clopidogrel, and they should be switched to a different medication. So we're trying to develop this assay that we will be running it daily if necessary, because the visit for PCI, the patients are either discharged the same day or the next day. So we want to provide those results relatively quickly. We still have a challenge of harvesting the volume and stopping the bleed. I think this is partially dependent on our education efforts and developing our new assay. I think that eventually we will be able to harvest most, if not all, of the pharmacogenomic testing needs. And then I'm working with several collaborators to build a research program around the pharmacogenomic testing, including smart sequencing, reporting pharmacogenomic results from our whole genome sequencing. We're working on the return of pharmacogenomic results to participants of large genetic research programs and several other smaller initiatives. So when we started this process, I had kind of in my mind, what is needed, to put together pharmacogenomic testing implementation. Unfortunately, all those elements were in complete disarray, and it took a while to put them together. And we also had to compete with external vendors. So I think right now we're in a much better place when we have, all those elements of successful pharmacogenomic testing implementation overlapping and talking to each other and interacting. And we've also got rid of significant proportion of the external vendor sending out testing. So I've been fortunate to kind of be a point person for the pharmacogenomic testing, but obviously it takes a village. Pharmacogenomics implementation is definitely a team sport, and it requires a lot of significant resources and serious institutional commitment. And we were fortunate because through Fairview Pharmacy we were able to engage with PMO process. That has been, this is part of the Fairview management process, that they've been very supportive and allocated some of the resources that they have towards the success of this implementation. there are lots of challenges from the selection of the technologies to the lack of reimbursement, which may be alleviated in the future. There is a bill that is going through the legislation that we've been monitoring and sending letters in support that may compel all the healthcare insurance companies operating in Minnesota to reimburse all biomarker testing, including pharmacogenomics. There is a huge need for education because PGX is not an absolute science. It provides opportunities for informed decision making, but it will not, replace the clinical judgment, and it will not always, be 100%. There are complex genetic variants that we already see in patients that we are testing that have unknown function, and we cannot provide, a specific guideline. So everything has to be taken with the sort of in the clinical context of the patient. And then there is an obvious need for active CDS. Pharmacogenomics is too complex for a busy clinician to perform ad hoc analysis or read multi-page PDF results. We have to streamline that process by building active CDS to help our clinicians and providers, and the most important, patients, to get the right medication based on their genetic makeup. So along the way, I've built a lot of collaborations that I want to highlight here, from MDL through College of Pharmacy, through Fairview Pharmacy. We work with billing, with EPMO, with genetic counselors, with Fairview IT, Institute of Health Mathematics. We've worked with North Shore, University of Florida, Mayo Clinic, CIPIC, and also I want to acknowledge the Minnesota Learning Health System scholarship leadership that allowed me to pursue that. I've been existing on Zoom for a lot of those meetings. A lot of that development happened during the COVID, essentially between 2020 and 2022. So during the height of the pandemic, so we've worked very well on Zoom, and I think I want to acknowledge the mute button on Zoom. It cuts down my natural sarcasm significantly, and that helps me with getting along with people much better. So I appreciate that. And then I also want to acknowledge Bill. Bill's been a very kind cheerleader of my efforts in pharmacogenomic development. If you wanna, he put together recently an update about the pharmacogenomic in more of a sort of better structured and layman terms. So if anyone is interested, it's on our departmental website, and I want to thank him for putting this little brief together. And he also put me on Twitter, so I think I can say that I made it in life, because I made it to Twitter. I don't have a Twitter account, but he put it there, so I appreciate that. These are some of my selected references that I used during that talk, and thank you for your attention. And we have just a couple of minutes for questions, if there are any.
Dr. Mroz?
Yes.
Hi, Vanessa Dayton here. Outstanding, absolutely outstanding presentation. How you took it from the basics to what you've accomplished. So, and now my questions. I have two questions. Could it be possible that the substantial excess, if you might say, of women, females, that the testing is ordered on has to do with the question about tamoxifen metabolism? Novartis is aggressively marketing their Femra, non-generic Femra, as the drug of choice for tamoxifen non-metabolizers. And that's CYP2D6 variants. And is that the possibility?
So that's why we put together the IRB to turn it into a sort of project, so we can actually get all all the patient data, and we do provide recommendations for tamoxifen on our report. So I think this is a quite viable hypothesis that the skewing towards the female population may be driven to some extent by tamoxifen. We're working on getting all that medication data, and so hopefully when we put together the paper in the coming months, we'll have a more specific response. But yes, it's entirely possible, and we do provide recommendations for tamoxifen.
And my second question is, speaking of marketing, is it possible that clinicians and or patients are opting for GeneSight because GeneSight does not require 30, 45 an hour, for the 30 minutes, 45 minutes, an hour of genetic counselor session for the patient, whereas apparently, is that still a requirement for everyone who gets this panel?
Yes, currently, well, I'm not, yes. So the way we build the system, and I think right now having a genetic counselor is very helpful, because the patients are not yet that much familiar with pharmacogenomic testing and its limitations, and it takes also away a lot of additional effort on the part of the providers, right? Thanks to having the genetic counselors, all the providers have to do is they just really need to submit the order in the SmartSet, because genetic counselors take care of sending the ABNs to the insurance companies, counseling and reviewing the data. So we have another set of eyes to look at our data. Well, it's difficult for the academic laboratory to compete with commercial vendors on several levels. You know, these are companies that are solely focused on pharmacogenomic testing. So they have people that do the outreach, people that develop assays, people that do marketing, etc., etc. Pharmacogenomic testing is it's only part of what we do here at the Molecular Diagnostic Laboratory. We have many other tests and assays, and we don't have dedicated resources to compete with those companies. Those companies were also first there, and they developed a specific way of simplified reporting when they created those green, yellow, and red bins that they put the medication. So the front page of the report sort of seemingly allows providers to easily classify which medications are good. So those are in the green bin, which ones may require those adjustments, which will be the ones in the yellow bin, and then the red ones will be the ones that are contraindicated based on the patient pharmacogenomic profile. Now the problem, so providers are very used to that kind of display, and we got some feedback from them like, oh, where are my green, yellow, and red bins, and we said like, well, this is not the standard. There is no standard across even providers which medications they put in any bins. This is just sort of a display that you got used to. This is not supported by CIPIC. This is not supported by AMP. This is just a marketing gimmick. But it's very difficult to wean them off that kind of approach in a way, and convince them that what we're trying to build here is a first step on the road to build the active clinical decision support that none of the commercial providers is able to offer. So once we get there, I think the argument of using a commercial testing providers will be irrelevant, because we will be just offering a much better approach to pharmacogenomic testing.
Well, Pawel, I have just a quick general question, kind of builds off Vanessa's. First of all, great job. This really was, you've done a terrific job here. And how would a patient even be aware that they would want to have this kind of analysis? I mean, how are they brought in? I understand the difficulties with payment and all this sort of thing, but is it the drug that they're going to be given that dictates whether or not they'll be tested, or who gets them into the portal, so to speak, to be able to, or how's that work?
So currently it will be the provider. So it will be the prescribing provider that may be aware that that's a very good question. So there are two sort of philosophies of pharmacogenomic testing that are currently being evaluated. One is a reactive testing, and this is the most commonly used approach. So the reactive testing means that the provider has a patient on a specific medication and the patient is not responding. So they had a trial of 2-3 weeks, and either the patient is not responding or the patient is not tolerating the medication. They set up another visit. The provider said, "Oh, why don't we adjust the dose empirically based on my experience? Let's cut it in half, or let's double it, or whatever it is." And they do another trial, and then the patient comes back and like, "Oh yeah, my symptoms, improved, but now I have a red face or whatever, or itchy fingers." So then the providers start looking for solutions, and I think there is enough now pharmacogenomic awareness that the providers know that there is that option for pharmacogenomics testing, so they would put the patient in. Now, the other philosophy that some of the leading centers implemented, like St. Jude, for instance, is preemptive testing, and the patient that walks through the door gets PGX profiling. Okay. In case in the future they will have a medication that may be affected by those results, and so we are currently in a very reactive process. What we're seeing is that's from conversations with providers. They usually have a trial of either different medications or different dosing before going to order pharmacogenomic testing, and this is part of one of the aims of the paper that we want to put together. We want to see those patterns. Sure. How over the year, whether providers were ordering it up front, or the providers how often they change the medication before they order pharmacogenomics testing, to kind of learn from that. I think with the active CDS in place, which is a goal for my next year and a half or so, is that we will try to build it to the point that the moment provider is trying to prescribe a medication in Epic, sure, if there is a pharmacogenomic result available for that patient, it will generate an alert. Sure. Or if this medication can be affected by pharmacogenomic testing, it will alert the clinician to the opportunity to test. Right. Sure. Absolutely. That makes sense. And then College of Pharmacy is building a website that is patient-directed about pharmacogenomic information. So hopefully we will try to link that website to MHealth Fairview website as well. So when the patients are looking or Googling for the pharmacogenomic information, that website will be popping up in this search. And this is in the layman language, patient-focused approach to explain the importance of pharmacogenomics. Thank you so much. Again, a terrific job. Thank you. Thank you. Are there any other questions? Okay, I think Amy left us. So if there are no other questions, thank you very much for your attention, and have a great day.